So if you're okay, Brian and Ahmad, I think we'll just get started.
Sure. Yep.
So welcome everybody to this barley community seminar hosted by the IBH. Today we have two speakers from Minneapolis. We have Brian Steffenson, who many of you will know, and Ahmad Sallam, who's one of Brian's colleagues. And the way it's going to work today is that Brian's going to start introducing this topic about the genetics of Fusarium head blight, and then Ahmad's going to take over. So very quickly, I say most of you will probably know Brian anyway. Were you originally from Minneapolis or Minnesota?
Just a northern suburb, yeah, just nearby the Twin Cities, yeah.
Oh, so I was just looking at what you sent us, Brian. I see you've gone kind of full circle from Minnesota to Australia to California to North Dakota, and then for the last 25 years or so, you've been back in Minneapolis again. So we've known each other for a long time. It's a real pleasure to have you here. I haven't known Ahmad for so long, but I'm sure he's going to give us a really entertaining and instructive presentation. So over to you, Brian, and yeah, on you go.
Okay, thank you very much for the introduction, Robbie, and good morning, good afternoon, or good evening to whoever's joining from whatever places in the world. It's actually a real pleasure for Ahmad and I to be invited to give a presentation to the IBH. And for me, it's wonderful to reconnect with the James Hutton Institute, because I had a very wonderful and productive sabbatical in 2010 to 11. So it'd be really nice to reconnect with everybody and see the new building the next time I'm in Scotland. So today, Ahmad and I are going to give a brief summary of our work on the genetics of Fusarium head blight resistance in barley. And I've worked on barley for, almost 32 years on various diseases. I think I've worked on 15 different diseases. This one by far is the most difficult, and I'll allude to why that is in the talk. So Fusarium head blight was first described in England actually in the 1880s, and from this map, the distribution is pretty much across all the areas where small grain cereals are produced. So only in areas where there's no cereals, or perhaps the conditions are so dry we don't have Fusarium, but most other places of the world it is found. And in places that are really warm and humid, that's where you'll see Fusarium being most severe. And as far as the causal pathogens, chiefly we have Fusarium graminearum, the major cause in our area. And this produces a mycotoxin called deoxynivalenol, or just abbreviated DON. Other people call it by its name vomitoxin for its emetic effects on swine when any of the Fusarium infected grain is fed to swine. So this pathogen has both asexual spores and sexual spores. The sexual spore stage is called Gibberella zea, and that's actually the spore stage that initiates the epidemic. So this fungus survives in residue, and then these ascospores initiate the epidemics. Now, there are other Fusarium pathogens, for example, POA, Sporotrichoides, and Avineasium, and each one of those produces a different mycotoxin profile. So just to give you an idea a little bit about Fusarium head blight or scab symptoms, usually at about the early dose stages when you'll start to see some kernels start to turn brown. And they go from anywhere from a light tan to a darker brown, while some of the other kernels are still green. So this is how you can easily tell that you have Fusarium. Now, there are other kernel blighting pathogens, but quite typically you'll see Fusarium causing this type of browning. And if the infection comes in really early, you can get complete blanking of the floret, so get no seed production. But if the infection comes in later, sometimes you won't even see much of a reduction in the plumpness of the kernel, but it still will have the fungus in it, and that can produce mycotoxins. Now, I don't want to get into too deep of plant pathology pornography, as I call it, but there are some significant pathogen signs that you'll see sometimes, and that is the black pseudothecia of the perfect stage that are shown in these arrows here. Now, as far as the effects on the crop, in some of the literature, we've seen yield losses exceeding 40% due to this disease. And that's, of course, a big concern to producers. But if you're a maltster, you're also concerned by the fact that this can reduce significantly the malt extract of the grain. And if you're a brewer, one of the worst things you can have happen is to have mycotoxins present in your finished beer and ales, especially something called vomitoxin. So there have been a number of studies where they've actually looked at finished beers and ales, and they have found traces of various mycotoxins. So this is a really big concern, and this is why the industry is really careful about not buying barley and malt that would result in mycotoxins being in the finished beer and ales. And in our breeding programs with Kevin Smith, he's really focused on low DON, low levels of mycotoxin in his breeding program. Now, the other insidious part about Fusarium head blight on barley is beer gushing. There are several things that cause beer gushing, but one of the main factors is there are hydrophobins produced by the fungus, and the carbon dioxide bubbles sort of gather around that. And so when you open up a bottle of beer, you can have things happening from just a little bit of over foaming to actual explosion of beer out of the bottle into your face. So again, a very big concern of the industry with regards to Fusarium head blight. So I started my professional career as a professor at North Dakota State University in 1989, and just shortly after I arrived, there was a widespread epidemic of Fusarium head blight. I had really no experience working with this disease at all. It seemed to have hit the Red River Valley area of Minnesota, North Dakota, and South Dakota quite hard. And the epidemic was so severe that many farmers just simply abandoned their fields. They didn't bother to harvest, or they would burn it if they could burn it. But the weather was so wet during that year that was very difficult to do. We saw yields drop from 92 to 93 by a very significant amount. And then they started to do testing for the mycotoxin DON, and they found that there was really widespread contamination of the crop. And then also there began the first reports of beer gushing for people who made malt and beer from the 93 crop. But strangely enough, during that year, the most impactful blowback from any group was somewhat unexpected, at least to me, and that was from the dog food industry, because a lot of dog food is using barley, and they were using Fusarium contaminated grain. And this was making the dogs vomit. And of course, this really got the owners upset, and they received 11,000 phone calls from upset pet owners. And then one company had to recall 16,000 tons of dog food. So that was an interesting effect of the epidemic back in 1993. Now, as far as the annual net revenue losses from 93 to 98, they exceeded over $400 million. And the epidemics continued after 1998. I'm just showing the data from 93 to 98. And we're continuing to fight this problem as well today. So you look at the cumulative losses to barley only, and they're very, very high in our region. And the epidemic was so severe and got people so upset that they got one of the senators from North Dakota, Byron Dorgan, to actually do a general accounting office audit about this whole situation. So you never want to have a GAO investigation, but that's what occurred after the epidemic. And then within the local paper in Fargo, when you make an editorial cartoon for a various issue, you know that it's a severe problem. And there were at least 2 or 3 editorial cartoons related to this disease epidemic. So something very significant in the region. And as a plant pathologist trying to solve this problem quickly, you know, it was a very helpless feeling, I think, in trying to help these farmers that were suffering so much from this disease. So when you look at the whole set of Fusarium head blight epidemics we've had over the last 2 decades, you look at the losses incurred by producers and end users alike in the supply chain. You look at the health risks associated with the mycotoxins and the use of infected grain. A lot of the human hardship and stress that's caused by the disease, as well as people losing their farms and their markets. I would have to say, and I think most people agree, these cumulative Fusarium head blight epidemics that we've had on both wheat and barley from this period of 93 to 2015 really rank among the worst disease-mediated agricultural disasters in the U.S. history. There's no question about it. It far exceeds the southern corn leaf blight epidemic in 1970. So my job at North Dakota State University when this happened was to try to come up with a solution. We did try fungicides with some mild success. They may have controlled the disease fairly well, but they didn't always lower the level of mycotoxin in the grain. So we did look at that. We looked at some different residue management strategies, but we also looked hard at trying to identify resistance. And this was a big learning curve for me because I'd never worked with the disease. So we were working on different methods to produce inoculum, both the sexual stage and the asexual spores, because we use those in two different inoculation techniques. In the lower left here, this is inoculation by my graduate student, Ray Page, and she's inoculating a conidial suspension on the barley varieties that were just at the heading stage at that time. So we had to work out all this method to make sure that we got good uniform disease so we could have a good screening procedure. The other thing that we did to try to speed up the process instead of having just one screening season per year is we connected with some Chinese collaborators at the Zhejiang University in Hangzhou, most notably Professor Bingxing Zhang. And sadly, Bingxing just passed away last week, so just really a sad bit of news. She was so instrumental in helping us develop these nurseries, and we got a lot of material screened through. We were looking at winter material and wild material, which we couldn't screen in the Midwest, so China was really important. And up here in the upper right corner are the three amigos, the barley breeders of Jerry Frankwick, Kevin Smith, and Richard Horsley that are out there screening in the Chinese Fusarium head blight nurseries. And you'll see that there's some bamboo posts sticking up amongst them. And the reason for that is that you cannot have barley lodge in a Fusarium nursery, because if it lodges even for one day, you'll get the disease overwhelming any variety. So we had to have the Chinese stake up every single barley, and we were screening thousands of them. So it was a lot of work to conduct a really effective Fusarium head blight nursery. And then unlike other diseases where you just simply score and then you're done, with Fusarium, you have to then harvest the grain, thresh it and grind it, and then submit it to the mycotoxicologist to get the GC mass spec data for the level of mycotoxins in the grain. Very expensive, very laborious to do. So it really separates it out from any other disease that we work on. Okay, so we did get off to a pretty good start in terms of figuring out how to obtain uniform screening nurseries and also to have the proper controls, because we need to know how the nursery is working and how uniform the infection is. So we had several varieties. This Chevron, the 6-row variety Chevron, had been known to have resistance for many years, even back in the 1930s. And it still is a standard of resistance for Fusarium head blight in a 6-row background. One of Kevin's lines called Stander is fairly susceptible, so we use that kind of as the, what we need to progress from what the current Minnesota reaction is for Minnesota barleys. CI 4196 is a Chinese 2-row that we use as their standard of resistance. And then finally, we had a 2-row susceptible. So these controls are really important. We put those in multiple replications to really understand how good the nursery was in uniformity. Now, this picture to the right, I would say is the very best that we can show as far as dramatic day and night reactions to Fusarium head blight. This one on the right here, this isn't a barley that's senescing. This is actually a barley that has almost 100% infection with Fusarium head blight. Why do I say that? Well, take a look at the foliage below. It's all still green. What's hitting the plant is actually Fusarium on the spike. So this is a really severe infection on this very susceptible variety. That's a 6-row there. And then on the left-hand side is a resistant 2-row or a fairly resistant 2-row. So this is how dramatic sometimes the differences can be between resistant and susceptible. Okay, so when we started this screening effort, we first started with the USDA collection. They have, about 30,000 accessions there, and we asked for almost all of them and started screening. But we also went to other gene banks to source as much diverse material as we possibly could. It was really fun to look at all the different types of barley with different 2-row, 6-row background, different colors and so forth. It was really fascinating to see all these being grown in the field. And we did extensive screening on all this material over a period of years. And I just want to give you a real quick overview of the results, which wasn't exactly great in terms of finding a lot of resistance. So after screening about 25,000 hordeum accessions, less than 0.1% exhibited what I would call partial resistance, meaning something close to chevron, but not often exceeding it too often. So, and then those that were identified in the very early stages of our screening process, they were selected to make mapping populations. So Ahmad will talk a little bit about that later on. And then of the ones that we evaluated, we selected out about 230 odd ones to do more intensive evaluations. We didn't want to throw out anything that might have resistance, so we selected these to have more intensive evaluations in multiple years and multiple environments. And the reason why this is Fusarium head blight interaction with barley is extremely variable. So you have to have a lot of replications, a lot of environments in order to feel like you have something real. And as far as the breakdown to that panel, it was about 43% were 6-rowed and 133 were 2-rowed, and we screened those in 11 environments. And I'll also add that during the time when we did the screening, this happened about 3 years ago, our industry went from traditionally 6-row type to now exclusively 2-row type. So that was a big switch when we already had a lot of material invested in a 6-row background, but now it has to all be in a 2-row background. The good news about that is that 2-row barleys generally get less scab and less DON than 6-row. There might be a number of factors related to that. We can talk about that later, but anyway, that's a good thing for us, I think, in moving towards the 2-row barleys. So this panel consisted of the whole gamut of development from old cultivars, land races, breeding lines, both feed and malting, Paulus types, and even some of the progeny from our mapping population. So these are all put into that selected panel for further evaluation. We also included some susceptibles for comparison, in addition to the controls that we had both in a 2-row and 6-row background, and of different heading dates as well, because that can be a factor in addition. So here's a quick summary of those results. So this is that panel of 234 barleys, and these are all the controls that we have from our super susceptible, this PI line here, to the resistant 6-row here with chevron, resistant 2-row 4196. You'll see that for Fusarium head blight, there were really only a few that exceeded chevron's level of resistance. And then with regards to DON accumulation, again, a very small number that were equal or better than chevron with respect to that mycotoxin. The other thing that was kind of frustrating for us is that we saw a fairly low correlation between Fusarium head blight and DON accumulation. So you basically have to look at both of them. Well, actually, in some respects, Kevin is really leaning towards just going on DON only and not bothering with having to rate Fusarium all the time. Just get the DON ratings and move ahead with the program. So this is a boil down to what we found for the 10 most resistant barleys from that panel of 234 and the 10 that had the lowest DON. And you'll see that there are ones from various mapping populations, there's some from various land races, interestingly enough, from Switzerland and Austria and some from Germany. But the thing that struck me very markedly was the ones with the lowest DON. So of the top 10 lowest DON lines, 5 of them were these S42IL lines. And maybe some of you might be familiar with that nomenclature, but these are lines from Klaus Pillen's introgression population. Klaus introgressed some genes from wild barley into the cultivar scarlet, and so 5 of the 10 came up as our top, lowest for DON. So hats off to Klaus for developing that and sharing that germplasm with us. And we've got a study now to try to map where those loci are conferring that low DON accumulation. But anyway, this is kind of what we're left with from that screening of the 234 barleys. Now, when you look at what the Midwest cultivar preferences are, we like to have a heading date of about 52 days and a barley that's not too tall, somewhere between about 72 to 76 centimeters. But if you look at some of these lines that we picked out, there's really only a few that satisfy that requirement. And then you look at something like the height, we have a lot of them that are just really, really tall. So the challenge is really to find resistant materials with the desirable heading date and height. And Ahmad will talk a little bit more about this, but we feel that some of the QTLs that have been identified, they seem to co-locate with various agromorphological traits. So the question is, do we really have true resistance loci, or is it just a pleiotropic effect of these agromorphological traits? So we did a GWAS for Fusarium Head Blight resistance, this actual severity here, and we identified 26 MTAs in this panel of 234 barleys, and this is based on the 50K genotyping. What we found is, as I just mentioned, that some of the Fusarium Head Blight MTAs co-located with heading date and other traits. So one very prominent one, of course, is we know about PPDH1, and we have a nice peak for Fusarium resistance right at that same one. So again, you have to be very careful that you're selecting resistance loci and maybe not some effect of an agromorphological trait. We also did the GWAS for DON level, and this is a lot more promising for us because we also identified 26 MTAs. But the great thing is that none of the big peaks seem to be at any known agromorphological trait. And we did study quite a few of them that Ahmad will talk about. So that's good news that we can hopefully move these loci into the breeding program and not just have it be due to heading date or height or something like that. So we took these MTAs that explain the highest amount of variation, were reliably identified in most environments, and were not co-locating to these agromorphological traits, and did the haplotype analysis to look at the diversity and effects in this barley collection, looked at the haplotype alleles for all these MTAs. And from this analysis, then, we selected 10 different founder parents, if you will. We had 8 that we could do, but we also had 2 in reserve. And so this table here shows the lines that we selected. So one of them was a progeny from Klaus Pillen's population, and there was a couple other from some other mapping populations that we had done. And then there were some land races from Switzerland and Romania, but also a couple of North Dakota and Minnesota breeding lines, so we don't lose all the malting quality in some of this poorly adapted material. So we went ahead and made this magic population in the scheme that you see here to the right. And we just finished with that in a grow out last season, and we've got it advancing 2 more generations. But we have 850 progeny from this magic population, and our first Fusarium evaluation will be this summer in 2025. So we're hopeful that something good will come out of it, but I'll tell you what, I've been burned before with this disease, so we'll see what happens. But just as a quick summary for what I've talked about here, From about 25,000 accessions screened of hordeum, we had a very, very low percentage that had what I would term useful levels of resistance. We did select this panel of about 234, really intensively evaluated that in multiple trials, which was really necessary to have the confidence in moving things forward in the breeding program. From the GWAS, we had 26 MTAs that were identified for both Fusarium and Dawn in the panel. And some were mapping to unique locations with no confounding agromorphological traits or lines. So that's good news. And then from that haplotype characterization I just talked about, we did identify these 8 founder parents for the magic population development. And as I said, I'm hoping that the accumulation of all these positive alleles for enhanced resistance will lead to progeny with enhanced resistance and also resistance to the accumulation of the mycotoxin, that we can pass these lines on to Kevin Smith that he can put into his breeding program and eventually make a good malting barley cultivar. So that's my portion of this talk, and it's kind of a lead up for Ahmad, and I'm going to let him take over the screen now and give his presentation.
So as Brian mentioned, that the resistance are oftentimes associated with agromorphological traits, and this is one of the challenges for working with Fusarium Head Blight and barley. For example, the resistance of Fusarium Head Blight is oftentimes associated with the row type, and there are 2 types of barley, the 2-row and the 6-row. And this this type is also, this phenotype is associated with linked to the VRS1 locus in chromosome 2H, where the 2-row barley are more resistant than 6-row barleys. Also, the morphology of the spike itself, lax versus dense, where the dense spikes tend to be more resistant to Fusarium Head Blight. Also for the seeds, there is the covered or halt or the hullus, and this phenotype is governed by the NUD locus in chromosome 7H, where the hullus barley lines tend to be more resistant to Fusarium Head Blight than the covered type. Also nodding spikes, where nodded spikes are more resistant to Fusarium Head Blight. Plant height, where taller plants tend to be more resistant because they are away from the source of the inoculum in the soil. Heading date is also associated with the resistance, where early heading plants tend to be more resistant to Fusarium Head Blight because they escape the window of infection later in the season. One of the other challenges for working with Fusarium Head Blight is the association with FHB resistance and an important multi-quality factor, which is grain protein content. In the chevron by M69 population, and chevron is a Swiss landrace that is resistant to Fusarium Head Blight and also resistant to kernel discoloration caused by Fusarium Head Blight and other pathogens. And M69 is a Minnesota elite breeding line. In this population, in 2003, the investigators identified a kernel discoloration QTL in chromosome 6H that explained large variance linked to a grain protein content. In the following year, they identified the exact same kernel discoloration QTL, in this case, linked to Fusarium Head Blight QTL, where chevron allele on 6H reduced FHB, reduced kernel discoloration, also increased grain protein content. And this is a type of repulsion linkage because the favorable resistance to FHB and KD is associated with the unfavorable high grain protein content. And high grain protein content is not favorable for malting because this results in the increase in the beer chill heads when the beer is exposed to lower temperature. So we don't know if these associations are due to linkage or pleiotropy. In the last 2 decades, more than 22 populations for Fusarium Head Blight were developed, and 14 QTL mapping studies were published for FHB resistance. And to better understand the genetics of FHB resistance in barley, we need to integrate all these QTLs together. But to do that, we need to develop a new barley consensus map because there is no single barley consensus map that has SNP, SSR, RFLP, and sometimes AFLP markers. For this reason, we constructed a new barley consensus map from 8 maps, including 6 linkage maps from previous QTL mapping studies, shown in the black color, and 2 previously constructed consensus maps, one for SNP markers and one for SSR markers. And we constructed this consensus map based on the linear graph theory, acting on the marker order and the positions between the markers. And the results is that we have a new map that includes 4,145 markers, including SNP, SSR, RFLP, AFLP, and morphological markers, and spans about 1,300 centimorgans. To look at the quality of this consensus map, we perform correlation between the consensus map positions and each one of the individual maps used to construct the consensus map. And we average all these values across chromosomes and across all the populations. And the average correlation is more than 0.95, meaning that we managed to integrate all those different markers together while preserving the order. So we performed a meta-analysis for Fusarium Head Blight Resistance in barley. And there are many sources of FHB resistance, but the most important ones are Chevron, is a 6-row landrace from Switzerland, and it's used in the Minnesota program in the early stages for stem rust resistance because it's a source for RPG1 gene. It also has a resistance to kernel discoloration. CIHO 4196 is a landrace 2-row from China, and it is the most resistant 6-row accession to Fusarium Head Blight. Undone. Gobernadora is a breeding line developed by Sebet Icarda in Mexico, is a 2-row type. Frederickson is a 2-row landrace from Japan. Kutaya is a 2-row landrace from Turkey. And Russia 6 and Zedar 2 are 2-row landraces from Russia and China, respectively. So to do the meta-analysis, we used the results from 19 biparental or triparental populations from 12 QTL mapping studies. And also we use the results from 2 GWAS studies that include 3 populations. And from this table, you can see this is the population size, the population type. Some of the populations advanced by selfing, some of them are double haploid, some of them are backcrossed, developed by backcrossing. And also the types of markers used in these studies, including RFLP, AFLP, SNP markers, and SSRs. In terms of the analysis traits that we use in this meta-analysis, that include Fusarium Head Blight resistance, down accumulation, heading date, plant height, spike angle, rachis node number, kernels per spike, and spike density. And these are the traits that were evaluated in many of the QTL mapping studies that were published previously. We collected data from those previous studies regarding the QTLs associated with Fusarium Head Blight, done and other agromorphological traits that include QTL positions and markers linked to those QTLs, marker variance, additive genetic effects, and the consistency of QTL detection across environments. So the final results is that we have total of 256 FHB done and agromorphological QTLs that were all projected into the new barley consensus map. This table shows the number of QTLs from the previous Fusarium Head Blight QTL mapping studies. In the first 2 row, we see that there are a total of 96 FHB QTLs were identified, and 57 done QTLs were identified in the previous studies. You can see also that chromosome 2H by far harbors the largest numbers of those QTLs, 43 for FHB and 27 for done. This figure shows a representation of the consensus map of chromosome 2H, and it shows the Fusarium Head Blight QTLs in black color and the done QTLs in red color. I'm showing also the consensus map location of important genes that are associated with important traits like PPDH1, controlling heading date, and VRS1 locus that controls the 6-row by 2-row type, and the CLY1, CLY2 cleistogamy gene that determine the opening of the florets. And we observed that all the QTLs in this area highlighted with blue are associated with PPDH1, the photoperiod response gene, and all the FHB undone QTLs in this region explains from 2.5 to 29.5% of the variation. All the QTLs for FHB undone in this area highlighted with yellow color, all of them are associated with heading date or height or both. And those QTL explains from 11 to 58% of the variation. All these QTLs in the highlighted by pink region are all associated with VRS1 locus. and they explained 4.1 to 71% of the variation. And all the FHB QTLs in this region highlighted with green are all associated with the CLY1, CLY2 cleistogamy gene, and they explained up to 26.1% of the variation. So you can see that most of the QTLs, or the important QTLs in chromosome 2H, are associated with agromorphological traits indicating pleiotropy rather than actual resistance. In terms of sources of favorable alleles for the FHB and non-QTLs, we could see from all the previous QTL mapping studies that chevron and chevron descendants that were used to validate some of the QTLs that were identified in chevron before, those chevron and chevron descendants showing blue contributed 28 FHB and 19 non-QTLs. This is followed by Frederickson that contributed 8 FHB QTLs and 2 non-QTLs. Gobernadora contributed 6 FHB and 4 non-QTLs, and then Zadar 2 and CIHO 4196. There are also that many FHB and non-QTLs explained low variance or identified only in a single environment. For example, 44 FHB and 28 non-QTLs, total of 72 QTLs, explained less than 10% of the variation. And you can see that from the frequency plots. No non-QTLs explained more than 40% of the variation. And very few FHB QTLs explained more than 40% of the variation. And those QTLs were either associated with either heading date or VRS1 locus. In terms of the consistency of QTL detection, we found that high number of the QTLs were identified in only one environment, including 65 FHB QTLs and 31 non-QTLs. So this meta-analysis demonstrated that large number of FHB and non-QTLs were identified. However, a few of them could be important for plant breeding using marker-assisted selection. And in plant breeding, we utilize QTLs that meet specific criteria. For example, QTLs that explain a higher portion of variance, like more than 10% of the variance. QTLs that are more stable, that are being identified in more than one environment. And QTLs that are independent from other agromorphological traits. If we apply these 3 criteria for all the previously identified QTLs, we could see that for FHB, we're going to have only 3 Fusarium Head Blight QTLs that could be used for breeding. The favorable allele contributed by Zadar 2 for 2 of them, and the third one, the favorable allele was contributed by chevron, and those are in chromosomes 2H, 5H, and 6H in barley. And for Dan, we're going to end up with 5 non-QTLs in chromosomes 1H, 2H, 3H, 4H, and 6H. And the favorable alleles for 3 of them was contributed by chevron, 1 from Rasmussen, and 1 from CIHO 4196. So from all the previous QTL mapping studies, you can see we went down from a total of 96 FHB QTLs and 57 non-QTLs identified in the last 2 decades, only 3 FHB and 5 non-QTLs that could be used for breeding. We know that selection using fewer number of QTLs that results in capturing only a small portion of the total genetic variation when we use marker-assisted selection. So genomic selection is an alternative approach because genomic selection uses a dense marker coverage to capture all the genetic variation of the population, including minor effects and major effects, and use this in the prediction model so the breeder could perform selection. Therefore, we expect that using genomic selection, we can increase genetic gain compared with marker-assisted selection. And in the Barley Improvement Program at the University of Minnesota, led by Dr. Kevin Smith, genomic selection is being used in the program. And part of my work, my Ph.D. work, I was assessing the accuracy of genomic prediction from several traits, including Fusarium Head Blight and Dan, for 5 independent cysts for 5 independent years. And we can see the prediction accuracy for Fusarium Head Blight ranged between 0.6 to 0.78, and for Dan ranged widely between 0.25 to 0.99. And we learned through this process that we need to increase the genetic relationship between the training and the validation sets to improve the accuracy of genomic prediction. In the breeding program, the breeder combines previous years to the current year to train a prediction model to predict the next year selection candidates. And then after making the selections and those progenies are evaluated and phenotyped, the breeder performed cross-validation, 5-fold cross-validation to confirm the accuracy of prediction for those progeny populations. And the breeding program doesn't use a single trait, actually used in genomic prediction, use several traits, including agronomic traits such as heading date, height, yield, lodging, and diseases such as DON for Fusarium Head Blight. The use is DON as the final product that mycotoxin found in the grains. Also for other diseases, including net blotch, spot blotch, bacterial leaf streak, and also uses malting quality factors in the genomic prediction. And these are the cross-validation values for the independent years. And something we could notice that the prediction accuracy vary from year to year, and this is expected because this is the result of the change of allele frequency and the change of the environments where the populations are being evaluated in. As a conclusion, there are no major Mendelian resistance or a major FHB QTLs such as FHB1 in wheat has been identified in barley. Numerous genetic studies identified many FHB QTLs with small effect that reduces the severity. However, those QTLs are not robust and not reliably effective across environments. And many of the FHB QTLs are co-segregate with a variety of agromorphological traits, including indicating pleiotropy rather than actual resistance. And QTLs for reduced FHB severity are consistently detected linked to the VRS1 locus, but only if the population segregate for the 2-row by 6-row type. And genomic selection is an effective approach to improve the breeding efficiency for Fusarium Head Blight resistance and DON accumulation. At the end, we would like to acknowledge all the collaborators from different programs and our research group. Thank you.
Thanks. Thank you, Ahmed and Brian. Are there any questions from the audience? There's a couple in the chat. Let me ask the first one, unless Uzal, do you want to ask the question yourself? Okay,
sure.
You ask on you then.
Yeah. So, yeah, thank you for the nice presentation. It's a good work in the area of Fusarium resistance. So I have a couple of questions, but my first question is like, do you know the origin of the reduced FHB locus in chevron?
Did you say the origin?
Yeah. Like how the chevron got this locus, like from who is previous, like the pedigree cultivar or breeding line?
So all we know about chevron is it was an old land race that was grown in Switzerland. So that's the original origin. So we have no pedigree information other than we know it's a land race collected from Switzerland and donated to the USDA way back in the early part of the 20th century, like in the 1920s or something like that. So that's all we know about it. It's quite tall. The notable thing about it, it has the brightest kernels. So in our industry, people like to have very bright kernels, and this has a very bright kernel characteristic.
Yeah. Okay. Yeah. Thank you. So my next question is, do you think like the PPDH1 is solely control the FHB resistance in barley, or there is a set of genes that stay together in the same locus and then control these traits? I thought, like I see, like the PPDH1 is highly focused in this presentation, and then it has a great effect in FHB resistance in barley. So that's why I'm curious to know about that.
Yeah, we often see some of the significant peaks for Fusarium and Don accumulation occurring right at PPDH1. That's most pronounced with the disease rating of Fusarium head blight. But the nice thing or favorable thing that we thought is that with regards to Don accumulation, and remember, that's the more important character that our breeder is interested in, we see that there isn't a co-location of the Don accumulation and the PPDH1. So that's good news for us in trying to identify loci that truly do reduce the accumulation of Don. I'm not sure if you want to add anything to that, Ahmed.
No, you covered very well, yeah.
Okay, yeah, thank you. So
Yeah, thanks.
should I move to the next question, or I'm sorry?
Yeah, sorry, I think I'm a terrible moderator. I'm going to ask Don if he wants to. Dom, not Don. I'm going to ask Dom if he wants to ask his question or comment, because I can't make out what you actually mean. So Dom, Vico.
Yeah. Hello, Brian. Just like if you want a non-dwarf PPDH1 stuff, it's actually the pure winter barley. So you guys are going in the right direction, escaping by making winter barley. That's it.
Ah, they are.
Australian are.
That's a great comment, and we believe that that's really the direction to go. So in our state of Minnesota, we have mostly corn and soybeans. I mean, even our wheat breeder says he's breeding a rotation crop for corn and soybeans. So barley acreage has gone way, way down. So Kevin Smith is now working on winter barleys. And a couple of decades ago, we thought that would have been just a foolish folly, but Kevin has developed lines that survive reliably in Minnesota now, and we believe that we're going to lessen the Fusarium problem by having the barley mature earlier, so we can escape largely the conditions when Fusarium is most prevalent and virulent. So good comment, and we're hoping to really show that winter barley can result in less scab. And I just wonder, in your experience, have you seen winter barley normally getting less scab in any places that you've worked?
I have to say, it does escape. Doesn't mean it's resistant, honestly. So it's then different. It's a different environment. So if you have the right fit between the disease and the cycle of the plant, let's say heading time, sounds like potentially the winter barley is taller, so we tend to have less, but we tend only, let's say in France and Germany. But I won't say it's out of experiment, it's out of farmer field. Spring barley to row, shorter dwarf, tend to be more sensitive.
Okay.
Brilliant. Thanks, Dom. And Miguel.
Yeah, thanks, Robbie, and thanks, Ahmad and Brian. Really nice presentations. In the Manhattan plots that you showed, Brian, in your part of your presentation, it seemed like for FHV you had a few MTAs with very high LODs. Maybe I saw them wrong, and also that in general you had larger LODs in the FHV side, right? Even you can see here in chromosome 6 you have a huge peak of almost LOD 11, which it seems very high also in chromosome 2, right, for FHV. And then when you see the Don ones, the LODs go back down and you barely have something that goes above 4, let's say, if unless I saw something wrong. So my questions were, what do you think you have these differences between FHV and Don? I realize it's not the same trait, but some sort of correlation I would expect. And the second question would be, do you think these huge peaks at FHV are still interesting to see, considering that you also said that it seems like Don is the trait to follow and not that much the FHV itself?
Yeah. Let me see if I can understand or repeat the question. Was that first part of the question, Miguel, related to the threshold of significance with Don and Fusarium? Was that it? Did I understand that correctly?
Yeah. So that in your previous slide in the FHV, you have a couple of MTAs, it seems to have a LOD of almost 11, right? In chromosome 6 and chromosome 2, unless I'm saying it wrong. And that for me is very high as compared to what you see in the Dawn Manhattan plot, for example. So I was wondering if do you think there's a reason for these huge peaks? And also, do you think these peaks are still interesting as to follow up or to pursue?
Yeah, maybe Ahmad, do you have any comments about that? I mean, certainly, you know, our measurements of Fusarium head blight, the way we do it is we have groups go out and count the number of infected kernels, and there's so much variation with regards to that trait, even across a single genotype. Whereas Dawn, we're actually combining all the seed across that row, and you know, with the GC mass spec, we're getting, you know, incredibly precise data. So that could be one of the factors. And then regarding the let's see, was it the Dawn or the FHB? Could we go to that slide again? Which one were you talking about, perhaps, Miguel? About, was it for Dawn or for Fusarium?
Sorry, in the
FHB. Oh,
yeah, and
going back to that one.
Chromosome 6, you have a green dot in chromosome 6.
Oh, okay. So this one up here, and maybe the. Yep. So we're definitely exploring, and I can't remember the haplotype analyses, but I'm certain that one of the parents that we chose for the magic population is considering capturing that haplotype allele.
That's great. And just a quick follow-up on this. Do you think the one in chromosome 6 is the same one, or the same or belongs to the same haplotype as the one Ahmad described at the end of his presentation? I think one of the reliable QTLs was in chromosome 6, if I remember correctly.
It's in close proximity, but not sure exactly. It is the ones from Chevron. It could be, but so GWAS in general is a way just to look overall about the significant associations, but this also should be followed by haplotyping and confirming. So and haplotypes will allow us to identify the favorable alleles and the individuals carrying those alleles. So this looks in close proximity to the grain protein and kernel discoloration, but it seems this section is a little bit toward the telomere, where the other section is more toward between 30 to 60 millimorgans on the consensus map. So it could be different, but I am not positive about that 100%.
Thanks.
So I had a question, Brian. I don't think there's anybody else put anything in the presentation. Oh, there is actually, Miguel. I've got that one. Yeah, so I had a question about the lines that you identified from Klaus Pillen's population. I presume they're all from the same cross. Is that correct?
Oh yeah, they are. So it's scarlet times Israeli wild barley spontaneum.
Are these from the Heb population? Is this one of the populations from the Heb lines, HEB lines?
Yeah, I think it's one and the same. Yeah, I'm not sure if Klaus or anybody from his lab, but I think that's correct. Yeah, it was a population of only like 65, and out of their 5 of their top 10 Dawn came from that small population. So yeah, it's interesting.
Yeah, I mean the reason that I was kind of interested in it because much of the presentation had the premise that this was a very quantitative, lots of minor gene effect, other than the developmental genes. But the fact that you've got 5 from that population in your top 10, which suggests to me anyway, and maybe wrongly, that the genetic basis of the resistance you're seeing, or the low Dawn in these populations, is either 1 or 2 genes. 5 out of the top 10.
Yeah, it could be. Ahmad and my technician are working on analyzing that data now. We have 3 years of data and multiple replications, so we're hoping that we can answer that question. But yeah, it would seem like it would be maybe just a couple of QTLs that are conferring that resistance from that small population. It's quite interesting how that came up. Those 5 came up to the top in the low Dawn category.
All right. And my impossible to answer question is, why would you get highest FHB resistance in Switzerland and Austria?
Yeah, it's a good question. You know, there's also stem rust resistance that's coming out of Swiss material. And a number of years ago, I went to Switzerland, I visited the gene bank, I visited the areas where these barleys came from, the old land races. And with regards to stem rust, the answer was very obvious, because there were barberries, the alternate host of stem rust, all over the Alps in this area where they were grown. So I would speculate that it was actually a coevolution of these land races to come up with several of these key resistance genes from Switzerland. Now, I could draw that over to Fusarium as well, that, you know, in the Alps, maybe the climate is conducive for Fusarium, and maybe the farmers through time made selections based on less scab and less stem rust. It's just pure speculation, but it's the best answer I could come up with.
Brilliant, thanks. Brian, I have to go, but I think Kelly or Malcolm can probably take over. Listen, thanks so much for your presentation. I'll speak to you again personally in a little while, but I have got a visitor just arrived at my doors.
Okay. Great, Robbie. Thanks. Good to see you.
See you later.
Bye.
Hi there. Yeah, I think there's just a couple of more questions and comments. Dom has some questions about Fusarium itself. Dom, you want to comment on that?
Yeah, just a question. We've been trying to practice as well those contamination, but at the end we were surprised that the race we were spraying were actually not the one killing the plant. Are you sure you which one Fusarium species is on your sample? Do you quantify them?
Yeah, so a little bit more about that I didn't have a chance to talk about. So each year, my colleague Ruth Domaki collects isolates from around the region. And then each year she prepares what we call the grain spawn inoculum and also the Canidia inoculum. And so she basically mixes a composite from the previous year as our inoculum each year, and it's all Fusarium graminearum. The reason that she does that is we have seen kind of a different pattern of these, what we call chemotypes. So there's some that produce only DON, some that produce some derivatives like ADON and so forth. So Ruth kind of hedges her bet to allow us to screen for what the current population of Fusarium is by making these collections each year and then compiling a composite, and that comprises our inoculum. So we're hoping that we, you know, don't lose something that's happening out in the field to the pathogen population. If that helps to answer your question.
Great, okay.
You have the collect sample. So yeah, there is diverse, is a mix from, let's say, you update the mix with the season, yeah. So you may have a different proportion of each species finally, or subgroup or race, I don't know what you call it. So maybe the effect is not like consistent pattern because you have various inoculum years to years.
Yes, that's true. But we sort of felt it was better to, you know, monitor the resistance to the current composition of the population than something, you know, from decades ago, because people have found kind of this divergence in chemotypes that has occurred in our area over time. So I'm not sure if it's always the best, very best strategy, but it seems as though the best resistant material, they are holding over time. So we're hoping and optimistic that, you know, it's going to be a stable resistance across whatever sorts of population composite is present in the field. But there's always a danger, of course, with the pathogen changing and, you know, some of these resistant types not performing the same as we've seen in the past.
Thanks, Rod.
Excellent. So there are no more questions in the chat. So this is last chance to anybody else. If they want to unmute or ask a question, please feel free. Right, I think sounds like everybody's has all the knowledge that they need from this. So I just want to say thanks again to Brian and Ahmed for their fantastic presentation, and if want to show your appreciation online, and this recording of this talk will be available on the International Barley Hub webpage. So thanks again for everybody turning up, and we'll see you in a couple of weeks for our next seminar. Okay, thanks, bye.
Yeah, thank you. Thank you very much for your attention.
Thanks.
I think the red X may be down here.